Good morning, students. Today we are going to see about the electrophoresis. So, electrophoresis is one of the instruments, that's the analytical instruments we are using in laboratories. So, this electrophoresis is usually used to separate the charged particles which are present in the substances or in the samples. So, let's see one by one. So, first we'll be seeing what is electrophoresis. So its literally means is to carry with the electricity. So with the electricity only we have to handle this electrophoresis apparatus. So what is the process which takes place in the electrophoresis means there will be a migration of the charged solutes. Charged solutes means it contains both the positive and the negative charges in the presence of the liquid medium under the control of the electric field. By applying an electric field, we are going to separate the charged solutes into one positive and negative. And that is a migration of the colloidal particles to the solution under the influence of a electric field. This is a electrophoresis. And this electrokinetic phenomenon was observed, and was first found out by a scientist called Rizos in 1807, who have noticed the application of the electric field caused the clay particles to disperse in the water and help to migrate. So this is the first process of electrophoresis who have found out by the scientist called Rizos. So many biological molecules we have ionizable groups, that is they contain charged substances in their body. So example like amino acids, proteins, nucleotides and nucleic acids, they contain charged substances. So under the influence of the electric field, the charged particles, that is both the negative and positive, will migrate according to the charges, oppositely charged substances. Like for example, the positively charged substances will move towards the cathode and negatively charged substances will move towards the anode. And the larger, that is the heavier particles, move more slowly than the smaller lighter particles. Say for example, if we have a nucleic acids, the smaller and the lighter particles of the nucleic acids will be moving fastly than the heavier part of the nucleic acids. So these are all the different types of electrophoresis, that is moving boundary electrophoresis, zone electrophoresis and isoelectric focusing electrophoresis. Today in this class we are going to see and focus about the zone electrophoresis. Under the zone electrophoresis we are going to see about the different types like paper electrophoresis, gel electrophoresis, capillary zone electrophoresis and immunoelectrophoresis. So let's start with the paper electrophoresis. This is a very simple technique usually used in the laboratories for separation of the small charged molecules like amino acids and small proteins. And here, as the name implies, paper, we are going to use a strip of a filter paper. So that is being moistened with the buffer solution. It's the buffer solution. It varies according to the type of the electrophoresis what we are going to use. And the end of the strips are immersed into the buffer reservoir containing the electrodes. And the samples are spotted at the center of the paper. So then the high voltage is applied by using a battery. So which makes that high voltage, which makes the migration of the charged particles according to their charges. And then at last the separated components can be detected by various of a variety of technical staining techniques depending upon their chemical identity. Usually, what is the type of paper we'll be using for this type of electrophoresis? Is a filter paper which is used for qualitative analysis, and cellulose paper, that is made up of 100% pure cotton. With this type of paper only we can able to handle this type of electrophoresis. and the paper absorbs the solvent, and it has to be at least height, they have to be 12 inches, and the strip of this paper, it has to be 50 centimeter. Let's see the diagram of the apparatus. So this is the paper electrophoresis apparatus. So these are the two reservoirs which contain the buffer, and at the center we have been placing a filter paper, and the two ends of the filter paper are immersed into the buffer, and at the center then we have to draw a mark, and that mark we have to apply the sample, that sample we are going to separate. And this buffer and so this apparatus is ready, and when we apply an electric field by using a battery, what happens, the sample will start to move according to their charges, oppositely charged substances, that is positive move towards a negative and negative move towards the positive. After this process is completed, we can take this paper out, and after the drying process we can make a staining technique, we can apply the staining technique, and we can able to found out the separated substances. This is a paper electrophoresis, a very simple type of electrophoresis. Next we are going to see about gel electrophoresis. Gel electrophoresis also, the process is almost same for all the types, but according to the type we are going to use. There we have used the paper, here as the name implies we are going to use the gel for the separation. So here also we are going to use a power source, that is with a voltmeter or voltage regulator, then electrophoresis tank that holds the buffer solution, then an anode and cathode which is connected with the power source, glass plate that holds the gel at the center and it's immersed into the buffer solution, a comb which is used to make the sample well in the agar. Agar is a gel we are going to use for this type of electrophoresis. Before it solidifies, we have to make a well, that is like a hole we are going to make, and we'll see that in the next following slides how we are going to make a comb. Then the voltage is supplied and it is regulated by the power source. So what happens, a current flowing between the anode and cathode to establish an electric field in the tank, which helps to separate the components, that is the charged substances. According to their charge they get separated, and according to their size also they get separated. That is we have already seen a small size of particles will move towards faster, and a larger size of particles which takes time to pass on to the separation process. So first, what is the first step? We have to seal the open bond. This is a plain glass plate, and we have to seal the four sides of borders and of the glass plate with the tape for forming a mold. Why we are making a mold? Because we are going to pour the gel liquid before solidification. We have to pour the gel. So here the second step, we are going to prepare the adequate volume of electrophoresis buffer. That is usually TB, which is called as TBE, that is tris borate EDTA, or TAE, that is tris acetate EDTA. These are commonly used to buffer, to fill the electrophoresis tank, and to prepare the gel. Usually the gel are in the powder form, that is agar gel, agar which is in the powder form, which we have to take in a correct amount of what we have needed, and that gel powder has to be made into the liquid by using this buffer solution only. Then added the needed amount of powdered agarose, that is typically 8 to 15% of the electrophoresis buffer, that is TBE or TAE, in a flask, Erlenmeyer flask. Then the buffer should not occupy more than 50% of the volume of the flask. Okay students, let's see the remaining part in the next class. Thank you.